So in this video, I'll be comparing DNA and RNA as a summary. I already made two separate videos specifically about RNA and DNA, so that is in detail. So make sure to check those out if you want a full, clear understanding on everything you need to know to get a really good score on your test. But for now, this will just be like an overall summary comparing the differences vaguely to make sure that you can confirm what you know. So first off, what's going to happen is first I'll explain the differences in function of DNA and RNA, like what about them do they do differently, like what's the different purposes. Then what I'll do is I have this little table here that I'll compare the three structural differences. How they look like, their differences. So three structural differences here, which I will reveal and explain. But up here will just be the functional difference. Okay, so what is the difference between RNA and DNA? So I already kind of used this analogy or this story to explain this, but I'm just going to do it again so you understand. Because I kind of explained this thing in the RNA video. So you know how, give me a sec. So you know how English is made up of alphabet, right? The alphabet, a whole bunch of letters. I think it's about 26. I'm not sure. But just like the English alphabet is made up of letters, you can arrange these letters to make words, and then you can add different words together to make a story, right? That's, for example, how a book is written, like Harry Potter. Now, RNA works very similar. RNA and DNA, they have their own little alphabet. So you can think, but their alphabet only has 4 letters, or 4. In science, you call them 4 bases, not letters. Now, if you arrange these letters that make up RNA, or you arrange these letters that make up DNA, you get a different DNA. So you get a different organism, a different characteristic, a different trait. Now, how does this idea of a book relate? So you can think of the difference between RNA and DNA is that DNA is like the entire book, whereas RNA is like a single page from that book, or a single chapter, a single section. Now let me elaborate to make this clear. Consider this door here to be a membrane. You know how your DNA is located within your cells, right? Specifically inside the nucleus. Now, inside this nucleus is surrounded by a membrane, a sort of wall, right? To make sure DNA doesn't just float out. Now, let's pretend this wall, this membrane, is locked or closed, and it has small gaps, like under the door, for example, that you can slip stuff through, okay? To transport things through the membrane. Now, our DNA is like our book, it's like our instruction manual. Everything about us is in there. How to fix us, how to build us, how to make us grow taller, how to make us look the way we do. So we know our DNA will be on the inside. So pretend this side of the door is your inside of your nucleus, and this side of the door is the outside of the nucleus. So inside the nucleus you have all your DNA, right? Everything about you. When something needs to change about you, say you need to grow, your body needs to find the information in the DNA. So it has to open this book, your DNA, and go find the specific information, the specific page on how to do it. But once it's found that information, and it knows where it is, it can't just bring out the book through the door outside the cell to go fix whatever needs to be fixed, or done whatever needs to be done, because this DNA is too big. It can't go through or under the door, because the door is locked, remember? So it can't go under the door. So how? It's not possible. So what it's got to do is it got to find the specific page and take that specific page. So say it finds the page that is responsible for a specific thing. It rips it out. Now it's good because now it can fit under the door, right? Now it can exit the membrane to go be done something with. But now that it's been ripped out of your DNA doesn't have it anymore. So next time the problem comes and it tries to find information, it won't be there because it was ripped out last time. So we're almost there. We know how we're going to get it through the door by having this page, but we want to make sure it's not missing from here. So what's going to happen is your body will go and photocopy this page. Okay, so it's going to go to a photocopier, not exactly a photocopier, but it will go to a place where it can copy this information. It's going to go there, it's going to copy that information, it's going to print it out just like this. So after you copy that page, printed it out, now you have that page and it fits on the door, plus your information is still in your DNA. You didn't rip the page out, you just photocopied it. So you can see that's the difference between RNA and DNA. DNA is your entire information, whereas RNA is like a specific page that was copied from your DNA, because your entire DNA can't fit under the door or through the membrane. But if you make a specific page copy, the RNA, it can transfer and go to wherever it needs to be done to go fix something. One small issue though, when it goes to copy this, your DNA, with the photocopier, in the copying process, something happens. You know how your DNA, I said before, it's made up of 4 letters. A, C, T, and G. Adenine, thymine, cytosine, and guanine. Those 4 letters make up your DNA, just like the English alphabet has 26 letters, right? So when it copies them, you can imagine this page will have ACTG, ACTG, just randomly put on this page, right? That's actually not exactly what happens. When it copies, everything copies correctly except one letter. One letter changes. And let me show you with this diagram here. So you can imagine when you go copy this book, you're going to get a page. The G will become a G, the A will become an A, the C will become a C, but the T, I'm going to make this T, but the T will not become a T. You would expect everything, all the 3, out of the 4 letters, 3 of them become the original one. Okay? So for example, you would suspect that T also becomes T, but it doesn't. T is the only one that doesn't copy correctly. It's as if there's like a mistake with the photocopier. Every time there's a T, it somehow puts another letter, okay? So instead of a T to a T, what happens actually is the T is made a U instead. So every time a T is supposed to be there, a U is there instead, okay? So that's what also distinguishes RNA and DNA. Their function is different, so they have a different purpose. One is a page, one is an entire book. But also their kind of alphabet is different. They have different bases. 3 of them are the same, but one is different. Every time it photocopies the DNA to make an RNA, everything copies correctly except in the RNA, you won't see a T. Every time there's supposed to be a T, you'll see a U. And U stands for uracil. But make sure to watch my RNA and DNA videos to make sure you understand all the bases and alphabet letters properly. Okay, so that's basically the functional difference. That's their difference in function. And I will go down to this table to quickly unveil what's happening here. So now in terms of structure, so now how the molecule looks like, okay? So DNA we know, we're familiar with, it looks like this double-stranded helix kind of shape. It has two strands. You can see the one strand, and then you follow the other one, the second strand, right? But then RNA is slightly different in the sense that it has a single strand. It's single-stranded. It's got only one strand. You can see there's only one thing you can follow. Whereas here, there's 2. There's 2 lines you can follow. Okay, that's one difference. And you can remember that because you can remember that RNA is like a copy of DNA, so it only copies a bit of it, a little section of the information, not all of it. Let's go to bases first. So we know the major differences in bases. Here is that DNA we know has A, T, C, and G, right? All those 4. But when the copying process happens, we know A stays A, G stays G, C stays C, but T turns into U. Like there's a small error in the copying process of the information. Okay? Lastly, sugar. What do I mean by sugar? So here's a DNA nucleotide. A DNA or RNA nucleotide. Let's pretend this one is a DNA nucleotide because it matches the column. And this is the RNA nucleotide. Remember, they're made up of 3 things. The base, the alphabet letter, the sugar, and then a phosphate. The last difference between RNA and DNA nucleotides, by the way, are what make up the entire RNA or DNA. If you put a bunch of these together, they'll make a string to make this kind of thing. You see, it'll make a string. But make sure to watch my videos to understand that, because I'm not going to explain it here. So the major last difference is this sugar. Although they look the same here, there's a small difference. They have a different name. These sugars are both sugars, but they're different types. So in DNA, it will be deoxyribose sugar, whereas in RNA, it will be ribose sugar. How on earth can we remember this? So we can remember that DNA has deoxyribose sugar because it's in the name. DNA stands for deoxyribo, that's for D, nucleic acid, deoxyribonucleic acid. So in the name, the beginning of the name that I put here in red, you can see deoxyribo, and that section represents the sugar that the nucleotides are made of, deoxyribose sugar. Now, it's going to be the same thing for your RNA. RNA has ribose sugar instead of deoxyribose sugar. You can understand that here because RNA stands for ribonucleic acid. So ribose stands for the sugar part. So both the beginning of these names, if you can remember the names, you'll know that the beginning of these names mean the sugars that they have. So they both have different sugars. So RNA's got ribose sugar, DNA's got deoxyribose sugar. Why do you think they need different sugars? So the reason why they need different sugars is because they have different alphabet letters, right? This sugar in DNA, the deoxyribose sugar, is going to bind to bases, right? The alphabet letters, specifically these four. But RNA's sugar is ribose, and it will bind to these four, right? So the reason why they have to be different is because they bind to different alphabet letters, different bases, okay? Because this structure is specifically made to be able to bind to those four specific bases, whereas this structure is specifically made to be able to bind these four. The deoxyribose sugar will never be able to bind a U. Its structure doesn't allow for that. And the ribose sugar will never be able to bind a T because its structure is different. It's not made to do that. Okay, so that's it. These are the four major differences: the function, the page in the book, the strands, single, double-stranded, the sugars that are different, which you can see in the names, and the bases that are different. And these bases are different due to the copying process that I explained above with the photocopier. The U and the T are different. So this is it for the basic summary. Make sure to check out the videos separately in detail. It will make you understand it even more. And I'll try and make a video next on some IB questions that you can come and watch and do with me to see if you understand.